Inside a police cordon, investigators prioritize collecting biological debris over searching for obvious weapons. They are hunting for the microscopic signatures of a human presence. At the absolute center of that microscopic search is deoxyribonucleic acid, DNA. Modern justice relies on the assumption that a single biological trace carries enough data to definitively clear a suspect or secure a conviction. That power comes from a mathematical reality. Except for identical twins, every human genome is distinct. While 99.9% of our DNA is identical, that remaining 0.1% creates over 3 million genetic differences between you and the person beside you. That vast variation has cultivated a public perception that DNA is an infallible mathematical certainty, capable of resolving any mystery with a single swab. And we leave that biological signature everywhere. The discarded coffee cup, a shed hair, a microscopic flake of skin, these everyday traces carry our complete genetic blueprint. To track those signatures, investigators rely on massive national databases like CODIS, which currently houses over 21 million distinct profiles, allowing local police to match crime scene evidence across state lines in seconds. When evidence is degraded or mixed, the tools get even more specialized. Analysts use mitochondrial DNA to extract profiles from ancient bone fragments, and YSTR profiling to isolate male genetic markers in complex sexual assault cases. We've built a highly sophisticated infrastructure to capture these biological breadcrumbs, but turning biology into a legal conviction requires human interpretation, and that introduces vulnerabilities the technology alone cannot fix. This photograph shows the environment where that interpretation begins. Far from the rain and mud of a crime scene, evidence is processing by technicians in highly controlled, sterile laboratories. The initial processing is clinical and precise. Analysts carefully collect the biological material, then use chemical extraction to isolate the DNA from the surrounding cellular debris. Because crime scene samples are usually tiny, they move to amplification. Using polymerase chain reaction, or PCR technology, specific regions of that isolated DNA are copied millions of times over until the sample is large enough to read. Finally, analysts examine over 20 specific genetic markers, called short tandem repeats. Comparing these markers against a suspect's swab generates statistical match probabilities reaching 1 in billions. It is a rigorous, demanding process, but its accuracy hinges entirely on one crucial factor: the assumption that the original sample was pristine to begin with. Real-world crime scenes rarely offer pristine samples. Evidence gets left in the rain, smeared in mud, or cross-contaminated by first responders. Even casual contact is enough to complicate a case. We now know that touch DNA, genetic material transferred by a simple handshake or brushed shoulder, can easily place an innocent person's biology at a scene they never visited. The math breaks down further during mixture interpretation. When a single swab captures overlapping DNA from 3 or more people, the statistical models become noisy and chaotic. Laboratories untangle these mixtures using genotyping software, but algorithms differ across jurisdictions, yielding conflicting match probabilities from the exact same sample. Then there is the human element. If an analyst knows the gritty details of a case, they can fall victim to the context effect, an unconscious cognitive bias that leads them to interpret ambiguous, mixed DNA results in a way that aligns perfectly with the investigator's existing theory. The intersection of trace transfers, complex mixtures, and cognitive bias transforms forensic profiling from a calculation into a subjective human judgment. This wood-paneled room is where scientific interpretations are finally tested against the rigor of legal argument. To prevent flawed science from swaying juries, judges demand DNA evidence meets the Daubert standard, a legal test requiring proof of valid scientific methods. It forces laboratories to defend their interpretation of complex mixtures and prove they actively mitigate laboratory error. To meet that standard, the field is realizing it needs outside help. As this presentation title suggests, neutralizing cognitive bias requires integrating forensic psychologists into the evidence review process to evaluate exactly how analysts are reaching their conclusions. True accuracy requires treating the human mind as a variable. By merging the hard biology of DNA with strict psychological oversight, we can protect the integrity of the results. Laboratories must implement structural firewalls, like sequential unmasking, ensuring analysts receive case information piece by piece to prevent bias. The system also requires independent oversight to audit practices and ensure genotyping software remains transparent and validated. Closing a case file represents the conclusion of an investigation that will determine a person's freedom for the rest of their life. We rely on DNA because we believe in its precision. But precision in a vacuum isn't enough to guarantee a fair trial. Evidence never speaks for itself. It must be interpreted. And justice is only served when we build rigorous, blind, and unbiased systems to do